வால்யூம். ஹலோ கேக்குதா? ஹலோ கேக்குதா? சார் 6 பேர் வந்தாச்சு மேடம். இப்போ நான் அட்மிட் பண்ண போறேன் மேடம். இப்போ நீங்க வந்து 6 பேர் உள்ள இருக்கான் வெயிட்டிங்ல இருக்கான். ஸோ வெயிட் த மீட்டிங் வில் ஸ்டார்ட் வித்தின் 15 minutes அப்படின்னு மாதிரி சொல்லுங்கண்ணா. யூடியூப் லிங்க் ஆமாம், அந்த எல்லா குரூப்லையும் ஷேர் பண்ணிவிடும். ஆமாம், இல்லை அதில் இப்போ ஷேர் பண்ணாண்டா சார். நம்பர் கூடின அப்புறம் ஷேர் பண்ணால் போதும். இங்கே 100 வந்த அப்புறம் ஷேர் பண்ணு. அட்மிட் பண்ணா இனி பேசாதீங்க.
ஆமா, அது எனக்கு இந்த இப்போ அனுப்பியிருக்கான். அது
பேசாதீங்கப்பா, கொஞ்சம் வெயிட் பண்ணுங்க.
This meeting is being recorded. The recording has stopped.
Dear participants, the meeting will start at 1 p.m. Good morning, sir.
Morning, good morning. Morning, madam.
Participants, please wait. We will start at 1:30. The participants will start sharply at 1 p.m. Good morning. We welcome all the chief guests of the sessions. Welcome, sir. Praveen, sir. Good morning. Praveen, sir. Good morning.
Yeah, good morning, madam.
Kaila, sir. Good morning.
ഇനി ആർക്കും വേണ്ട CSF ഇല്ല ആയിക്കഴിഞ്ഞാൽ നമ്മൾ നാണക്കേട് തന്നെ CSF വേണ്ടിയിട്ടുള്ളൊരു കോളേജ് സിവില് വേണ്ട നമ്മൾ മുഖത്തടിച്ച പോലുമല്ലേ പറയുന്നത്
Sroka. Sroka. The Kailash. Sir. Yeah.
കട്ടിയായിരുന്നു ഞാൻ കുടിക്കാൻ പറ്റിയില്ല കളഞ്ഞു ഞാൻ പൾസ് പോലെ
Good morning, ma'am. Anna, ma'am. Good morning. Ma'am, can you hear me?
கேக்குதா?
என்ன சார்? நான் தான். அஸ்வினி மேம் வந்து சீஃப் கெஸ்ட் இன்ட்ரோடக்ஷன்.
ஹலோ மேம், நான் அஸ்வினி. கேக்குதா மேம்?
ஆமா அஸ்வினி மேம் கேக்குது.
ஓகே மேம்.
Anna ma'am, good morning. Can you hear me? Ma'am?
Good morning.
Anna ma'am, can you hear me? Ma'am?
சிக்னல் கிடைக்க மாட்டேங்குது. அதான் சிக்னலே கிடைக்க மாட்டேங்குது.
Hello. Hi everyone, this is Praveen.
Okay, good morning, sir.
Yeah. So, Professor already joined. Because Bragos also joined. Hi. Hello, Anna.
What, sir?
Hello. Yeah, thank you. Thank you for being here. And I think they will start
Okay, sir.
few minutes.
Morning. I'm Jesse Pius from Rogini College of Engineering and Technology. It's my pleasure to welcome all to the International Conference on Emerging Trends in Material Science organized by Department of Civil Engineering, Physics and Chemistry. Nothing is impossible with our God as long as we have faith for His mighty power. First of all, I'd like to invoke our Almighty to shower the showers of blessings. Let's start with the Tamil Thai Valthu. The glory of Kanyakumari lies in the confluence of three seas, wherein the shore, the famous Bhagavati temple, and into the sea, well-known Vivekananda Rock and the gigantic Thiruvalluvar statue are there. But our Rogini College of Engineering and Technology add another feather to the glory of Kanyakumari. Our college is nestled in a lush green, pollution-free campus and a spot of beauty encompassed by canals, paddy fields, beautiful gardens, which provide us with breezy, lovely, and lively environment. It's a vast campus with an array of trees, brims with pride on the excellent infrastructure. In short, our college is gifted with the ornaments of nature. Welcome every morning with a smile. Look on the new day as another precious gift from our Creator, another golden opportunity. A smile is the universal welcome to the pleasant moment. Now I welcome Dr. S. Radhika, Head Department of Chemistry of Rogini College of Engineering and Technology for the earnest welcome address.
Thank you, ma'am. A warm greetings from Rohini College of Engineering and Technology. A dream written down with a date becomes a goal. A goal broken down into steps become a plan. A plan packed by action makes your dreams come true. With this note, I would like to thank each one of you for joining this international conference on emerging trends in material science. ICETMS-21. I feel great proud to say that this conference has been organized by our Civil Engineering Department in association with Department of Physics and Chemistry for over 8 years and has chronicled huge success each year. Our RCET management organizes such conferences, enlighten the young minds, and promote the participants of students, research scholars, and faculties. I am profoundly delighted to take an opportunity to welcome our most respected chairman, Sri K. Neelam Markandan, Honorable Managing Director, Dr. N. Neelavishnu, great source of knowledge, our beloved principal, Dr. R. Rajesh, who took out their valuable time and joined us today to be a part of this international conference. We are honored to have you all with us. Even though this conference is the marker of success of our institution, it also allows us to meet some of the brilliant minds of the country, which become the chief guest for this occasion. With all those words, I am extremely delighted to welcome our chief guest, Dr. Anastroka Batnika, University Professor, Independent Unit of Spectroscopy and Chemical Imaging, Chair of Biomedical Science, Medical University of Lublin, Poland. Next, I would like to welcome our resource person, Mr. Gregor Scolis, a research scholar, Medical University of Lublin, and Mr. Praveen Narayana Perumal, researcher, scientist, Medical University of Lublin. We are delighted to have you among us. On behalf of RCET management, principal, head of the department, faculties, research scholars, students from RCET and other institutions, I would like to extend my warmest welcome. I would also like to offer my regards to our head of the Department of Civil Engineering, Dr. Sahaya Ruban, and Dr. Jesse Payas, head of the Department of Physics, coordinators, and the faculties who made this conference function in such hard times. Welcome the challenges, look for the opportunities in every situation to learn and grow in wisdom. I express my hearty welcome to my dear students, research scholars, and faculties to make this ceremony worthwhile. I guarantee that the conference will be productive and worth your precious time. Once again, I welcome you one and all.
And the wonderful administrator and the high achiever of the institution, he is Dr. Rajesh, to give the presidential address. Sir, please.
Thank you, ma'am. Respected chairman, respected managing director, respected chief financial officer, the most honorable chief guest of the day, Ms. Anna Stroga Bartnika from Poland, and various resource persons, the convener of this program, Dr. Sahaya Rupan, Professor and Head, Department of Civil Engineering, and the Head Department of Chemistry, Dr. S. Radhika, the Head Department of Physics, Dr. Jesse Fayes, my colleagues from Department of Civil, Department of Chemistry, Department of Physics, and participants. Very good afternoon to all. So it indeed is a great pleasure to participate at the International Conference on Recent Advances in Material Science. It is jointly organized by our Department of Civil, Chemistry, and Physics. So I would like to appreciate that steps taken by our department colleague, Dr. Ruban, Dr. Mr. Sudan, Mr. Rajiv Gandhi, Ms. Ashwini, and all the faculties of Department of Civil, and Dr. Radhika, Dr. Jayanishubha, Dr. Ms. Sugandhi, Ms. Anita Kumari from Physics, Dr. Jesse Fayes, Professor Victivale, Ms. Tanga, Ms. Wesley from Department of Physics, and all my faculties for their painstaking effort to organize this conference on behalf of Rogani College of Engineering and Technology. So I am very much delighted to welcome in a special way that Dr. Anna Stroga from Poland University. That she is a very good researcher and in the field of FIT or imaging and Raman imaging. And her contribution is very immense. Just 5 minutes before, I had gone through her publications. Really, it is very good. Very good. And we are very much happy to have a wonderful resource person on this day. So on my behalf and on my the behalf of the Rogani College of Engineering and Technology, I extend my warm, special welcome to Dr. Anna Stroga Bartnika, madam. Please accept our welcome, ma'am. And I am very much happy to welcome Dr. Grace-Ra Hailas. And he had made a lot of contribution in the field of spectroscopic analysis. And Dr. Praveen Narayana Perumal. And he has also done wonderful contributions in the field of material science. So my dear friends, definitely this conference will provide an opportunity, will provide a platform for the research scholars, for the policy makers, for the academicians, for the industry people in the field of material science. So when we are talking about the material science, that lot of research has been going on day by day, month by month, year by year. Lot of researchers are publishing lot of work and lot of patents also coming. But still now we didn't find the hardest material rather than tungsten or diamond. And similarly, we didn't find the softest material like a talcum. And okay, anyhow, but people have developed a lot of materials which are being used in our aircraft, and even in a small, tiny pin to very huge aircraft. A lot of research work is going on in the material science, and scientists have developed lightweight materials with very good stiffness, very good toughness, very good hardness. Okay, anyhow, but we could not be able to find out the hardest material than the tungsten or diamond. Okay, so with the help of through this conference, I hope that this conference will pave a way to find out that softest material and as well as the hardest material. And apart from this, nowadays, material in the material science, lot of research opportunities are there in the field of the medical field. Medical field, lot of opportunities are there, and similarly, in biomedical field, also a lot of medical opportunities are there in the agricultural side, in the food science, in the chemical side, very good opportunities are there. So the researchers, the participants who will attend these conferences definitely will get an ample opportunity to expose, to exhibit their hidden talents, to exhibit their papers, to exhibit their research findings, and get the opinion, get the contributions of that our very good researchers like Dr. Anna Stroga, Batanica, and other people who are participating in this conference. Once again, I appreciate the organizers for organizing this wonderful conference in our institute for the betterment of our researchers. So thank you for giving me the opportunity.
Thank you, sir. Now I request Mrs. R.K. Ashwini, Assistant Professor, Department of Civil Engineering, to give the chairperson introduction. Ashwini ma'am.
Hello? Hello, ma'am. Is it
Yeah.
audible? Hello.
Yes, yes.
Oh. Hello, everyone present here. It's a great honor for me to introduce the guest speaker, Dr. Anna Shroka Badnika, chemist, passionate about spectroscopic imaging techniques. Anna ma'am did her PhD in Poland at Polish Academy of Sciences in the field of mass spectrometry and solid-state NMR of biodegradable polymers. Currently, Anna ma'am, she is a professor, chair of biomedical sciences, Medical University of Lublin, Poland. It's a great privilege for us, ma'am, to have you with us. Thank you, ma'am.
Thank you, ma'am. Now I am very eager to invite our special invitee, Dr. Anne Sharoka Batrishai, University Professor, Chair of Biomedical Sciences, Medical University of Lublin, Poland, for the chairperson address. Welcome, ma'am.
Good afternoon, dear participant. It's really great pleasure for me to welcome all of you at this 2021 International Conference of Emerging Trends in Material Science. It's really a great opportunity to exchange our experience on investigation various materials involving biomaterials, polymer science, polymer materials, metal complexes, nanoparticles, and nanoscales materials. So different backgrounds often leads to developing interesting idea and bridling questions in the discussion, which is the definition of emerging science, setting new trends, methodologies for the future development. So this is the innovation that is very often looking from a different perspective for the materials, and it may lead to many moments of inspiration. And I wish you that this meeting will be very fruitful for your future career, and we can exchange our knowledge and get new ideas. So during these conferences and in the future events. Thank you.
Thank you, ma'am. Thank you.
Thank you, ma'am.
இனி இப்போ எப்போ போயிட்டு எப்போ வரும்?
It's time to thank all the dignitaries. Good afternoon to all. I thank our Almighty for His presence and guidance throughout this inaugural session. It's my privilege for giving the opportunity to express the gratitude on this special occasion. Thanksgiving is the time of togetherness and gratitude. First and foremost, I extended my wholehearted thanks to our renowned chairman, Shri Kenila Martandan, who is the center source of support. Then I thank our dignified principal, Dr. R. Rajesh, who extended his value and moral support to conduct this conference. Then I thank our valued chief guests of the sessions who are going to spare their time in spite of their busy schedule. Today we are going to hear the thoughts of them. Then I thank the organizing secretaries, Dr. J. Sahaya Ruban, Professor, Head Civil Department of Rohini College of Engineering and Technology, and Dr. S. Radhika, Head Department of Chemistry. Then I thank all the HODs of various departments. I thank all the coordinators for making this function grand success. I'd like to thank all the respected representatives who have blessed us with their presence. Then I thank all the dignified peoples who are just sharing this session for showing their interest in these sessions. Thank you one and all. Now we can start the session one. Now I call Mrs. Ashwini, Department of Civil, to introduce the chief guest of the session one. Okay. Sir, we can start this session one. Madam? Ma'am, you can start the first session.
Microphone.
Good morning. Can you see my presentation?
Yes,
Yeah, we can see.
Yeah, okay. So thank you everyone for very nice introducing, and it's a great honor for me to present my research for you. The title of my talk is The Use of ATR, FTR, and Raman Imaging to Evaluate Bone Formation on Biomaterials of Ceramic Type in Vitro. So this part of the work was done at Medical University of Lublin with my all collaborators, which I will indicate them during my presentation. So, sorry. First of all, why this topic? So as you know, this is the challenge for the biomedical and engineering technology to produce the new biomaterials, there is a lot of needs to repair the bones during the injuries, during the cancer treatment. There is still a lot of things to do in this biomaterials science. The bone substitute biomaterials, they should be easy to prepare, easy to handle in surgery. They should be bioactive and biocompatible. Why it's so important? Because this is during the surgery where the biomaterials must be implanted, and it must be very easy and useful and sometimes flexible to put into the body to make this easy during the operation. But nowadays there is the new trend there are 3R experiments, which is the development of method to replace, reduce, and refine animal experiments. Oh, sorry, something. So nowadays, we don't want to use animals in the lab. And even in the biomaterial science, we are, sorry, something is interrupting my presentation. So let me close it. Yeah. Can you see my slides?
Yes, ma'am.
Yes.
Yeah. Okay. So okay. Sorry for this. So nowadays there is the new idea to avoid using animals, or sometimes when we cannot avoid using animals, we want to reduce the use of animals. Why? To reduce the cost, to reduce the shortened time of the analysis, and just because of this ethical problem of the using animals in the lab. So what is the new idea for that? The new idea for that is concept of using tissue engineering products, that we can produce biomaterials and then put our isolation, our stem cells or cells from our body, put onto biomaterials, and then do the implantation of this tissue engineering product into our body. So this is the future plan of what the perfect biomaterials in my idea should work. So this is a feature from my colleague paper, Dr. Agata Przykora-Kuśnierz. So this is what we can do, take from the blood isolation the stem cells, prepare the in vitro studies, and then load the stem cells onto the biomaterials and then do the implantation in the body. So this is the new idea which the whole scientific world is going to work in this direction. What is this? What is the spectroscopy methodology that we can participate in this idea? So we would like to develop FTIR imaging and Raman spectroscopy imaging technique to investigate that kind of biomaterials after in vitro studies, to analyze the lipids, carbohydrates, protein. We can analyze the degree of mineralization process, and very important fact is to analyze the maturity of the collagen. So in our study, we work with in the collaboration with Dr. Agata Przykora. She is my colleague from the department, from the Medical University of Lublin, and she is expert in the producing the biomaterials, which we use 3 components, which is chitosan, beta-glucan, and hydroxyapatites. So first 2 components are polysaccharides, which are connected with the hydroxyapatite. Why? Those polysaccharides make the hydroxyapatite structure very flexible when it's wet in the water. When it's dry, it's very rough materials. But when it's in water or in the fluid, then this biomaterial becomes flexible. That is very easy handle for surgery. So in our study, we would like to present different approaches, different spectroscopic methodology to analyze the biomaterial, which is based on these 3 component analysis, like chitosan, beta-glucan, hydroxyapatans, and then we're setting with the stem cells. For those experiments, we use 2 kinds of the stem cells. First is ADSC, adipose-derived stem cells, and the second cell line is bone marrow derived stem cells. So we use those 2 kinds of cells. So what was the design of this experiment? So we used the stem cell setting and incubation for 21 days. We used the control material, which was just cell medium without the cells, and we put the biomaterial in the cell medium. The next sample, we use just pure biomaterial, which is free component based on chitosan, glucan, and hydroxyapatite. And the next sample was we use biomaterials, we put the stem cells on, we load the stem cells on it. So they were, the cell line was apodized derived stem cells. And the next sample, to compare different cell lines, we use another cells, which were bone marrow derived stem cells. And our method. So we got 4 samples: control sample, native sample, which is just pure biomaterial. A control sample is biomaterial incubated in the cell medium, but without the cells. And we have 2 biomaterials, ADSC and BMDSC, which was the biomaterials loaded with the stem cells. And we will, during the analysis, we will discuss these differences between those 4 samples. Our method. In first part of my presentation, I would like to present the ATR-FTR spectroscopic imaging. Then I will focus about the Raman spectroscopic imaging and DREV spectroscopy to analyze those 4 samples. So very shortly, what is the vibrational spectroscopy? As you know, is the interaction of the light with the matter. So this is my favorite definition about that. And when we talk about molecular vibration, you know that the atoms are in the movement. We have different vibration that could be seen both by the Raman or by the infrared spectroscopy. So first paper, I would like to present my collaboration with the Professor Sergei Kazarian. That was part of the research was done at Imperial College London in United Kingdom. So we use macro ATR imaging to analyze our biomaterials. On the ATR crystal, we put our biomaterial, and the crystal was attached with the FPA detector. So the imaging was done on the whole surface, which was connected with the crystal. This is very, maybe I will shortly tell about the imaging system, that they are not single spectroscopy, but they are several spectra which are connected in the XY directions, and they consist on the group of spectra, and those group of spectra we connect into the images. So during that experiment, we used the micro ATR FTR imaging using the gold ligand, the IMAG, which is the SPECAC accessory, and was connected with the microchamber and FPA detector. That equipment was used at the Imperial College London. So to present the first data, first we concentrate on the coloration about singles, our pure materials, which was chitosan, beta-glucan, and hydroxyapatite. So we see the differences in the spectra and the bands which are in different direction. Here are these indicating points which we use for the fingerprint. So we can even notice the difference shift in the main peak bands of the hydroxyapatite. Then we analyzed the ATR FTR sample spectra, which were native, control, ADSC, and BMDSC. You can see the different lines, and we can compare that the lines with the ADSC and BMDSC, they are with the stem cells, and the native in control, those are the experiments were not used cells. So adding the cells is make a difference in the shifting bands, even in the lipids and even in the amides, of course, which is quite obvious. And next part that we use for the analysis were images. So we put the biomaterial on the crystal, then we image the surface, and this surface was imaged by 0.6 times 0.55 millimeters. So this is the area of the image. We use the color scale to express the high concentration and the low concentration, which is the intensity of the bands in the map. And what we can notice that there is different distribution of the lipids in those native control and biomaterial samples. And we also notice, we can also observe different distribution of the amides band. So that was done example. Going to the other components that or substance that we can analyze with these biomaterials. This is we can analyze carbohydrates, phosphates, beta-glycosylic bonds, and there also we notice different distribution within the biomaterial samples. So using that kind of accessory, it was used that we can not only use for the single spectroscopy, and we can see the differences between the sample, but also we can look at the distribution on the surface. So this is very important information for that, especially when we would like to look at the lipids, amides, and carbohydrates, which indicates the intense cell of the proliferation of the cells which were incubated on the biomaterial surface. And this is the most important part of this work, which is the collagen. We can assess the collagen maturity. So we choose two samples which were covered, which were with the stem cells. There were two lines, ADST and BMDST, and we measured the mature collagen and immature collagen. And as you know, there is the difference in the bands shift in the ATR spectra, and then we can present the ratio. So having that information, we can predict which stem cell line will provide to the faster proliferation of that on the biomaterial surface. Our results based on ATR imaging were also confirmed by confocal laser scanning microscopy. So we use another technique to confirm our study. So type 1 of the collagen was here on the left column. And on the right column, we have the osteocalcin, which confirmed that the cells really grow up at the biomaterial surface. So those are the examples and confirmation of our technique. Next part of my presentation, I would like to focus on the Raman imaging. So here is my team members, my group, and some nice castle of the Lublin. And that part of the research was done in Lublin. So biomaterial, which was hydroxyapatite.
The slide is not visible.
Okay, sorry, I don't know why. Let me. It's okay right now?
Okay.
Can you confirm?
Okay, we can see.
Okay, but you follow the preview. Yeah, okay, great. So right now there's the Raman imaging is the control sample that we can see the granules, and the organic matrix, which is present on the right corner here. So this is with the green and blue colors. So we would like to focus how the organic matrix is distributed between the hydroxyapatite, and we also would like to present the difference in the hydroxyapatite after the incubation in the medium and stem cells. So to do that, we use the second derivative of the spectra, Y, to notice the very small differences in the, for example, shifting the peak of the hydroxyapatite. So from those samples, from these points, we know that this is the hydroxyapatite structures, and we use how the spectrum looks like in the middle of the granule on the border. Part is from the polysaccharide matrix. So just to notice if the incubation of for 21 days in the cell medium can influence the structure of the hydroxyapatite, and how much. And of course, we use the distribution on different maps, this is the, we focus here on the polysaccharides to note and how they are changing. Going deep there, we move to the bigger areas of the Raman imaging. So we choose different part of the sample. As you know that the biomaterials are porous materials. They are differences in the surface, and so the structure is different. So we choose different parts like granules, the parts between the granules, and what happened, what is exactly between the granules. So for that, we also use not only showing the distribution of the three components like hydroxyapatite, polysaccharides, chitosan. We would like to know what's going on the surface, on the border between those two organic and inorganic matrix. Next, we choose also bigger area. This part was also published that we choose a bigger area to image to notice if there is also some small parts or bigger parts. And what we focus here, we would like to see how the polysaccharide matrix, how it's rebuilding during those samples, native, control, and those both with the stem cells. And we would like to notice also if the what happened is when we mix them, if they are a mixture of they are separate or how they behave each other. So that images allow us to analyze the distribution within the samples. Also, we would like to focus on the hydroxyapatite band, if they are shifting or they are moving or they have some subbands. So we use the control sample and ADSS samples to have the different distribution of the samples. And but that was the analysis about the surface. But what happened in the deep of the samples? So we use the deep profile. So those are the deep profiles of the samples going from this line profile into the deep of the sample. So measure each layer by each layer to have the distribution of the samples. We would like to notice what is on the surface and what is below. If there are some prediction that one of the substance would be on the top, which substance would be on the bottom, or there is no, we would like to test what's going in those the 21 days incubation in the medium cells, if there is change. The next part of the presentation, I would like to focus infrared spectroscopy
Ma'am, and the voice is breaking. It's not audible. Ma'am?
In the first part of the presentation was the contact with the crystal. The second part was the Raman imaging, and third part is drift. So it's a reflectance from the surface of the biomaterials. So we use our four samples, the native, so the control, which was the pure biomaterials, and two samples which were incubated with the stem cells for 21 days. So we noted the differences in the in those peak and those band. This is the subtraction of those bands with this line, and we would like to see the difference in the lipids, carbohydrates and phosphates, what's between them samples. And also we notice the different distribution between those two samples which were set to the steps. So going to the conclusion, those different application of the spectroscopic imaging allow us the identification what's going on the biomaterials seeded with the stem cells, which composite could be visible on the surface, and we can measure the maturity of the collagen. And also we showed that ADSS cells showed increased synthesis of the type 1, which was the higher extracellular matrix mineralization. And those three techniques, spectroscopic technique, were very useful for analysis of these materials. And now I would like to thank to all my team members, the Dr. Agata Przekora team, which were responsible for production of the biomaterials and setting the stem cells on it, and my team, spectroscopy team, that we were analyzing using different technique and analyzing those biomaterials. This study was funded by the Foundation for Polish Science, that we got funding for that. And I would like to thank also my collaboration from Imperial College London to Professor Sergei Kazarian, and from Warsaw Institute of Physics, Dr. Isabella Pienta. And also we work with the mass spectrometry part within this project from Stockholm University, Dr. Leopold Tilak. So thank you very much for your attention. And this is a nice picture of Lublin, our city in Poland.
Thank you, ma'am. Thank you. Participants, if you're having any queries, you can ask.
Yes, ma'am. Hello. Hi, ma'am. I'm Dr. Tijani Shubha, and I would like to ask a question, which is what is the advantage of ATR FTIR method over other spectroscopic methods for the analysis of biomaterials?
So for me, using the ATR spectroscopy is like a passion, and what is the advantage that we don't need to destroy the material. So the most powerful technique, which is like macro ATR, that we put the biomaterial on the surface crystals. What does it mean? You know that the biomaterials, which were in this case were with the hydroxyapatite and polysaccharides. So hydroxyapatite, they are granules, and they are different size, like 200 micrometers of the granules. So you know that they were porous surface of that. So that's why the biomaterials are quite difficult to analyze. And for me, using the infrared and Raman spectroscopy, or you ask about ATR spectroscopy, is that we don't destroy the sample. We just put them. We don't crush them. We don't have to cut the sample in the small pieces. We don't have to make the structure, the physical structure of this biomaterial is not destroyed. And after the analysis, we can take the same sample. For example, we can do, because those are non-invasive technique. So even if we put the sample, it's just take a contact, we don't destroy the sample. So that was in my opinion, the advantage of using that kind of samples to analyze. Of course, we support our study with different techniques like confocal microscopy. We use the SEM, scanning electron microscopy analysis to be sure and confirm our data. So this is from the analytical point of view, we usually use 2 or 3 different analytical methods to confirm our results. So, but the main tool for us is the infrared and Raman spectroscopic imaging.
Okay, ma'am. Thank you. Thank you so much.
Thank you.
Ma'am, myself Radhika. How the Raman and IR spectroscopies differ from DIFT spectroscopy?
Hello.
Ma'am,
could you repeat the question because I didn't hear you.
Yes, ma'am. How the Raman and IR spectroscopies differ from drift spectroscopy?
How they are different from drift spectroscopy. Yes?
Yes. Yes.
Hello? Yeah. Okay, so why we use drift spectroscopy? It's a very good question because the drift spectroscopy, it just penetration of the surface, and we use like diffusion. So it's diffuse. It's not. The main idea is not when you put not flat sample on the flat surface, yes, you have the porous samples. So sometimes you have the contact with the crystal when it goes into ATR crystal. But using diffuse, that there is the penetration on some depth of the sample. So when we have stem cells and we have different porous or rough surface, we would like to notice what's on the cover or what's on the edge of the samples. That's why we use drift if we were able to notice something more. Yes, if we were able to notice differences between the samples if those differences are on the same level that could be noticed by Raman or ATR. So that was also our idea to correlate different vibrational spectroscopic method in investigation biomaterials, to have an idea if, for example, hydroxyapatite we can measure by Raman, which would be, and for example, infrared, which is more sensitive for lipids and amides. So we would like to notice and use all our technical and analytical methods to do our best to analyze biomaterials.
Thank you, ma'am.
Thank you.
Can you give some more explanation about stem cells?
So the stem cells were done by Dr. Agata Przykora-Kuśnierz. So she's an expert. They are not so easy to work with them. First, you need to work with the cells. They need to do the incubation. And then it's not so easy to load them onto biomaterials. We did several tests, and it was not easy to find the good condition to design our biomaterials that the stem cells want to grow on its surface. So as you know, the science is not easy. So it really works a lot, needs a lot of tests. And that part of this work was done by Dr. Agata Przykora-Kuśnierz, who is expert in setting stem cells. If your stem cells wants to grow on it or not. So it's difficult study but it needs a lot of work experience with that.
Okay, thank you, ma'am. Any more queries? Then one thing I would like to ask is you have shown some graphical representation, that is a wave number versus the absorption. How we are getting the output? How we are getting the graphical representation of the wave number versus the absorption?
How can we, could you repeat the question?
You have shown some graphical representation, that is it's noted wave number in the x-axis and absorption in the y-axis. So how we are getting the output? What is the method actually used there?
But you mean about single spectroscopy or about images?
Yes. Single spectroscopy.
About single spectroscopy. So the single spectroscopy, so we can compare the spectra between different, like the intensity of the band. You mean what kind of first we look at the fingerprint of the spectra. We would like to notice if they are lipids, carbohydrates. Then we look at the subbands if they are. Sometimes we do the convolution of the spectra and the intensity. And some of the peak, if we would like to present changes in the like amides part, we very often normalize the peak by the phosphate band. And then we can notice the changes which are more sensitive in the amides band, for example.
Okay, ma'am. Thank you.
Is the
Ma'am, one more question. What is the advantages of using confocal laser scanning microscopy, ma'am?
So we would like to confirm our data with different technique, and we use confocal microscopy. So we use two parallel set of the sample. One set of the sample with the biomaterial with the stem cells, and that part was used for infrared and Raman spectroscopic analysis, and the second was used with the confocal. So, but using confocal, you need to use chemicals to your sample. You need to dye the sample. So you need to make colors, your collagen, osteocalcin, and it's more expensive so it's wet technique that you need to buy the sets, apply to the samples and do. But both methods are very available. So that's why we use we try to apply different method to make sure if we really see what we see in our images.
Thank you, ma'am. Any more queries?
Ma'am, can we use the ordinary scanning electron microscopy for tissue engineering products analysis?
Yes, of course. It's one of the basic technique that is used for this tissue engineering products. Depends of you what you do you want to see. Yes, so always designing the science for me is like choosing the proper technique for the samples. So if you would like to first confirm that your stem cells are really there, it's worth to use the confocal laser microscopy to be sure and confirm the collagen, for example, maturity, and if there is the osteocalcin, and so the proliferation of the cells goes well. And after that, we did the infrared, so we were sure that our stem cells are really there, and they are on a surface.
Thank you, ma'am. Thank you, ma'am. Thank you. Now I call Dr. Jani Shubha, Associate Professor, Department of Chemistry, to introduce the chief guest for session 2.
Thank you.
Thank you.
Thank you. Thank you, Dr. Jessie Pius. Good afternoon, ladies and gentlemen. It is a great pleasure for me to introduce our speaker of the session 2. He's going to talk about spectroscopic analysis of Kerdian and chitosan polymer matrices. Let me now give a brief introduction about our guest speaker. Our speaker is a PhD scholar at the Independent Unit of Spectroscopy and Chemical Imaging at the Medical University of Lublin, Poland. He is a 2015 graduate in veterinary medicine, currently working on his PhD by investigating cell biomaterial interactions with spectroscopy and microscopy. He is an author of 20 publications and a participant of internships at the universities of Naples and Pisa in Italy and at Stockholm University of Sweden, and a member of the International Society for Clinical Spectroscopy. With this brief introduction, I invite our speaker, Grahosh Kalish, to take over the session.
Thank you, Anna ma'am. Now we invite the next chief guest, that is Grahosh Kailash, for second 2. Welcome, sir.
Good afternoon. Hello, everyone. Thank you for invitation. I would like to greet all the participants. First question, am I heard right? The sound is good?
Yes, sir. Yes.
Okay, so I will start sharing screen. Can you see my presentation?
Yes, sir. Yes.
Okay. So again, hello. My name is Grzegorz Kalisz. I know it's kind of difficult to pronounce it, and I'm almost. Sorry. What's the problem? Can you hear me?
No, sir. No, sir. You can proceed.
Okay, so I'd like to present the results on investigation of Curdlan and chitosan Curdlan hybrid matrices, molecular structure and properties. This is all evaluated, investigated with Raman spectroscopy and FTI spectroscopy, because I'm a part of Dr. Anna Sroka-Bartnicka team, and also with cooperation with Dr. Agata Przekora-Kuśmierz, our independent units of spectroscopy, chemical imaging, and tissue engineering and regenerative medicine, and also physical chemistry in Warsaw. So all of that will be a result of gelation temperature change. But briefly, we can start about what is Curdlan. This is a natural polymer made of glucose monomers, of D-glucose monomers linked by glycosidic bonds. They are mostly exclusively beta 1,3 glycosidic bonds, and it has unique gel forming properties, which lies in the name from the word curdle. In higher temperatures, higher than 50, it's forming gel, quite stable, and it's very useful because it's non-toxic, flavorless, elastic, biodegradable. So it has a variety of applications, usually in pharmacy, medicine, food industry. We'll focus on our material part, so within medical and pharmaceutical discipline. So our aim of the study was evaluating properties of Curdlan gel in two different temperatures. It is, and the second one was chitosan. So it is also naturally existing polysaccharide. And in our experiment, we used the krill-derived substrate from C. Fisher Institute in Gdynia. It is composed of randomly distributed beta 1 to 4 linked D-glucosamine and N-acetyl D-glucosamine substrates. It is produced commercially just by the acetylation of keting. It is also commonly used in pharmaceutical industry, in the medicine or food industry, as a drug carrier, in tissue engineering for regenerative medicine, and for food packaging. It is biodegradable, bioadhesive, and non-toxic. So its usefulness is also very high, and moreover shows anti-cancer, antimicrobial, and antioxidant and anti-inflammatory activities. In our research, we focus mainly on Raman and FTI spectroscopy, and it's about inelastic scattering of photons, named after Nobel Prize winner. We use just laser-induced vibrations with 532 nanometers laser, putting it in virtual energy state and later measuring the radiation. FTIR is different in principle because this is about infrared light absorption by chemical bonds, and we used Fourier transformation to acquire spectral results. And the third one, spectroscopic method is XPS. This is more surface sensitive and quantitative technique used by irradiating surface area with soft X-rays and measuring the kinetic energy and the number of ejected electrons. And our first experiment was on Cardinal polymer matrix. It was published 1 year ago. We prepared in water bath in 2 different temperatures, which we have chosen 80 and 90 degrees. This was also prepared by Dr. Agata Przekora-Kuśmierz. So after gelation for 20 minutes, films were were cooled down, dried, and acquired films were subjected to Raman, IR, XPS, and to microscopic techniques, AFM, which is atomic force microscopy, and scanning electron microscopy. And coming to results, in Raman, mean normalized spectra were quite similar. Here are presented in plane A the most distinct change, which is change of intensity in the range of 200 to 650. To investigate it better, we performed second derivative, and second derivative is shown in plane B, revealing 2 new bands attributed to a skeletal distortion and to ring vibration of carbohydrates in our sample gelled in 80 degrees. In other ranges, no notable changes of spectra were present. We can see here ranges between 1200 and 1500 with second derivative, and we can see that the spectra are quite similar. So here you can see point with arrow 487 and 494, which is the most notable change. Similar approach was with ATR, FTIR spectroscopy. So it showed similar pattern for both samples, but second derivative in plane B and C revealed many additional bands in 80 degrees sample comparing to 90 degrees sample. They were assigned to changes in secondary structure. No other changes were observed. So as you can see in spectra and second derivatives in planes D, E, and F. So here are the most important changes. In XPS wide-scan core level spectra for carbon, oxygen, and nitrogen, we performed the convolution of peaks, and it showed the surface functionality changes, which was increase in wrapping mostly of carbon bonds, also in sample in 90 degrees. It was consistent with results acquired from nitrogen and oxygen peaks. So this information indicates on higher density of glucan molecules after gelation in lower temperature, chosen from these 2. And finally, microscopic images from atomic force microscopy and scanning electron microscopy present surface of samples recorded, which in 90 degrees is more rough comparing to 80 degrees, because this is why, because the fibers are more densely packed and this wrapping of carbons is happening. Similar observation was made with scanning electron microscopy. We can see higher granulation in 90 degrees sample, as you can see at the bottom of the slide on the right side. Later, we decided to perform similar research on Cortland chitosan hybrid matrix. It was also published, but we decided to choose different temperatures. They were more intermediate, 70 degrees and 80 degrees. Also, the procedure was similar. Gelling of Cortland happened for 20 minutes in different temperatures. Chitosan needed to be gelled chemically with sodium base. And also thin blended films were spread now on cover slips and subjected to analysis with ATR, FTIR, Raman spectroscopy, and microscopic techniques I mentioned before. So firstly, I'm showing the results from FTIR spectroscopy. Sorry, I forgot to write a note. But in FTIR, the disappearance of shoulder bands was found. In plane A, you can see our substrates we investigated before going to our blended film to indicate indicate specific bands to these two polymers. And in planes B and C, you can see our blends investigated in 70 degrees and 80 degrees gelation. The most notable changes were at approximately 3060 and 10 and 3085. They were found in chitosan, and it indicates on interaction between an amine group of chitosan and presumably hydroxyl group of our glucan in our sample of 70 degrees. So more bonds was formed in lower temperature. This effect was also revealed by absorbance intensity ratio, for example, between amide1 and amide2, contributed to carbonyl and amine stretching vibrations of amide groups. So it also can be used to determine the changes in secondary structure of polymer chain. To investigate it even further, more information was acquired from second derivative spectra of amides 1, 2, and 3 to monitor changes the intensity and resolving broad overlapping bands into individual bands, resulting in the accuracy increase. So beyond slight shifts, there is band loss at 1577 and appearance of two new 5079 and 76. Variations in the spectral range means changes in secondary structure. The alpha helices content decreases and the protonation occurs, which suggests different chain folding, cross-linking temperature of 80 degrees. So this higher temperature. At this figure, you can see that each band is assigned to maximum and percentage share surface of entire surface area. Observations were also made in Raman spectroscopy, but FTIR brought more information. Raman spectroscopy indicates the same manner from amide range between 1200 and 1300, indicated on change in secondary structure. And finally, atomic force microscopy. We investigated the surface to see that on the surface of our 80 sample, some bulky items were observed with different height comparing to 70 degrees. So 70 degrees sample has more smooth surface. and topology of 80 sample is more rough with these bulky items. Those are functional groups, it's good. Sometimes it depends on what we desire from our biomaterial, because sometimes it might be good to have points to absorb proteins or even activate, promote cellular response just by having these domains to catch cells. Depending on what we want from our designed product. So to conclude, carbon gelation temperature affects distinctly molecular arrangement, surface structure, and topology of polymer, which can be modified in production regarding application. And it goes with solely Corlan or even hybrids made of Corlan and chitosan. In 80 degrees, compared to 90 degrees, it has higher density, higher stability, and lower porosity. In our hybrid, the minor secondary structure were confirmed between intermediate temperatures, 70 and 80 degrees. And as I mentioned, the presence of different active groups on the surface may promote specific protein absorption, affecting cellular response to the produced biomaterials. It may be useful in this probably wound management, but already mentioned biomaterials used in bone substitution also. And the most important for us was that cooperative use of spectroscopic and microscopic approaches allow evaluation of products gelation in different temperature. It is very useful because at the stage of developing biomaterial, we can evaluate its properties correctly even before putting them to animal testing, to human testing, just by using spectroscopy and, for example, cell incubation. So it's very, very promising for making all the research and development faster and cheaper, by the way. So thank you for your attention. You already met my supervisor, and I would like to thank very much for her supervision, because it gave me a lot to think about and a lot of experience. And for the whole team, we are working together on this project on biological chemical imaging parts. The cooperation is going great, so it's nice to be a part of this team, and also for Foundation for Polish Science, because it allows us financial support. And that's all for my presentation. If you have any questions, I would be happy to try and answer them. So please feel free.
Participants, if you're having any queries, you can ask.
Generally, we use polyethylene terephthalate, polyvinyl chloride, polypropylene, etc. These type of polymers are generally used in food processing. What is the main attention of using this Curdlan in food industry?
Well, I think it will be the most important to address this gel forming, because it's unique for the whole industry, and differing result. We already knew that that depending the temperature, the properties of Curdlan will be different. So that was not the point to investigate. Our point was to investigate how it changes and what is changing in which temperature, because this information was not yet investigated. These properties with chitosan, it is widely used in pharmaceutical industry, and people are trying all over the world to put different kind of drugs and apply it with, for example, in ointments, in stuff which is injected, not injected, but edible for pharmaceutical industry. We already knew that this hybrid is beneficial in promoting cellular response after many years of experience from Dr. Przekora and Dr. Soroka Bartnicka. So that's the point we use these specific polymers. If I got your question right, if you are happy with my answer.
Thank you, sir.
Sir, what is meant by this polymer matrix?
Sorry, what is? Can you repeat the question?
What is meant by the polymer matrix?
What is the polymer matrix?
Yes, yes.
Well, it's I mentioned that we made thin films. The polymer matrix is what we call specific, very thin layer of polymer, or solely only cordlan or cordlan chitosan, and this thin layer can be put into some application. For example, you can make it in plasters and put on the wound as a cover. It's very elastic, so it covers the wound great, which is, for example, preventing from microbial microbes or even other damaging factors. And this layer can be also put in other parts. So if we try to commercialize it, for example, it may be used, it will be just called matrix or some different name, but matrix for now is sufficient enough.
Thank you, sir. Any other queries?
Sir, is cytosine is a synthetic polymer?
This one we used was not synthetic, was more natural, because it was acquired from krill, from Sea Fishing Institute. So it was kind of a natural one.
It's a polysaccharide, correct, sir?
Yeah. Yes. Yeah, it's a polysaccharide.
So the matrix formation, like the cordlan and cytosine polymer matrix formation, is easy or somewhat difficult? The formation of the matrix.
Well, this question is more for Dr. Agata, because she was responsible for developing this and fabricating it for us. We were more focused on spectroscopic evaluation, but after initial problems with acquiring good quality of products, after working with chemicals and temperature, it is not anymore very difficult, because as you saw, the procedure of fabricating biomaterial is for cordlan just gelation in water bath. So for 20 minutes, you just need to set the temperature right, mix it, stir it correctly, and later cool it down. So you will acquire good product. And with chitosan, there is an additional step to chemically gel chitosan, but it's also not something very difficult. So after just knowing how to perform the production methodology is quite simple.
Equal 5,000 riyals.
Thank you, sir. Any other queries? Okay. Thank you, sir, for your wonderful explanation on the topic. Now I call Mrs. Rajalashmi, Assistant Professor, Department of Civil, to introduce the chief guest for session 3.
Thank you.
Okay, thank you, sir. Ma'am, please.
Thank you, ma'am. Hello, everyone present here. I'm honored to introduce speaker Dr. Praveen Narayana Perumal, scientist, the Department of Biopharmacy, Medical University of Lublin, Poland. He pursued his PhD under the supervision of Professor N. Raman at Madurai Kamaraj University in 2016. He worked as a research scientist for 1 year. Now currently he is working as a scientist. His research focused on the department of multi-target directed pharmacos for neurodegenerative disease. And he also received several awards. And his research achievements are he published 20 research articles in the previewed reputed journals such as European Journal of Medical Chemistry, Journal of Neurochemistry. It is a great privilege for us, sir, to have you with us. I invite Dr. Praveen, sir, to present a title, Metal Complexes as a Decompositing Agent for Environmental Hazards. Welcome, sir.
Thank you. Can you audible? Can you
Yes,
hear me?
sir.
Yeah. So can you able to see my slides?
Yes, sir.
Yes, sir. Yes, sir.
Yes, sir. Yes, sir.
Thank you. Thank you so much for giving me a nice introduction about me. And I would like to extend my thanks to the managing board of Rohini College of Engineering and the organizers of this international conference, Dr. Sakaya Ruben, Dr. S. Radhika, and Professor N. Sudan Kumar. Today I'm going to talk about metal complexes as a decomposing agent for environmental hazards. So environmental hazards. Environmental hazards has developed by so many factors. Among them, one is pollution, the second one is global warming, and third one is deforestation, and the fourth one is soil erosions. So today I am going to mainly focus about pollution, especially water pollutions. So water is easily able to pollute, especially by contamination of chemicals. So para-nitrophenol is one of the chemical substance which is extensively used in the industrial field and as well as act as a pesticides in the agriculture field. So despite of their importance to the usage, it is highly toxic substance to the environment that exists industrial and agriculture wastewater in abundance. Most importantly, it has been classified as a priority toxic pollutant by the US Environmental Protection Agency. So this is the chemical structure of so this is the chemical structure of para-nitrophenol. The nitro group is presented in the para position of phenol. On the other hand, para-aminophenol, it is one of the chief intermediate in the preparation of numerous analgesic and antipyretic drugs such as paracetamol and acetylidine and so forth. Also, it is acting as a strong reducing agent to develop the photographic films. Also, it is used as a corrosive inhibitor in paints and anti-corrosive lubricant agent in fuels for two-cycle engines. In the dye industry, para-aminophenol is used as a wood stain and converting a rose-like color to timber and as a dyeing agent for hair and feathers. So the chemical structure of para-aminophenol is instead of nitro position, there is the amino group, which is NH2. So is it possible to convert para-nitrophenol to para-aminophenol, which is a toxic compound to use the environmental and human beneficial drugs? Yes, is it possible to convert using the reduction reaction in the presence of some catalyst? In this regard, so many catalysts are developed by scientists, nanomaterials, photocatalysts, and quantum dots. But these all materials have some limitations. For example, photocatalyst, is it very difficult to reusable the catalyst. And the nanomaterial, the surface of nanomaterials is not enough smoothie, so the rate of reaction is very less. And most important thing is all these materials are energy consuming materials. So in recent years, metal complexes are used as a decomposing agent or a catalyst for a reduction of these kind of toxic toxic elements. Before that, I would like to give you a brief introduction. I would like to give a brief introduction about metal complexes. I feel that most of the participants from the engineering field, so I would like to just recall the basic chemistry, which is metal complexes. So metal complexes is formed by two chemical entities such as ligand and metal ions. So ligand is usually as an organic one, which is able to donate lone pair of electrons and acted as a Lewis base. And metal salts are usually acted as a Lewis acid and able to accept the lone pair electrons. And when it goes to the complexation, the metal complexes will be formed. And compared to other all materials, metal complexes have several advantages in the para-nitrophenol reduction reactions, which are highly thermal stable and large surface area, which help to the energy transformation in the catalytic process. And most of the metal complexes are poor water solubility, so it can be help to. So that is so we can able to perform the reaction in a water medium, and metal complex is not soluble in water, so it is very easy to recollect and reusability by simple filtration technique. So we have designed and synthesized a few copper-2 complexes, copper-2 mixed ligand complexes using tube base as a carrier ligand and dicarboxylic acid as a co-ligands. So the synthetic protocol of the preparation of complex is the orthophenylene diamine is reacted with two equivalent of benzaldehyde in presence of ethanol to form the tube-base ligand, and the tube-base ligand is further reacted with metal chlorides and dicarboxylic acid to form the corresponding metal complex. And the metal complex is structurally characterized by various spectroscopic techniques. This is the IR spectroscopic technique, IR spectroscopic data. So you can see this data, in around 1600 to 20 centimeter inverse 1. That is corresponds to the CH double bond N stretching frequency. This frequency, when it goes to after complexation, it goes to downfield. It's due to the chelation. So this spectroscopy is confirmed that tube-base ligand and complex formation. In order to further confirm, we have recorded the mass spectroscopy. The overall molecular weight of the complex is 449, but we got the molecular and peak at 450, which is M+1. So mass data is well matched with the calculated one, which it is confirmed the complex formation again. So this compounds is failed to grow the crystals. So the electronic paramagnetic spectroscopy is one of the best spectroscopy to prove the geometry of the compound and the bonding nature of the complex. So you can see here the compounds are based on the gyromagnetic ratio of free electrons, gyromagnetic G parallel and G perpendicular values. You can predict the geometry is distorted sculptural geometry, and the G value, which is gyromagnetic, which is axial symmetry, which clearly state that there is no exchange interaction in the compound, so the compound is mononuclear in nature. So after careful structural characterization, we have performed the catalytic reaction with para-nitrophenol. So the reaction mixture contains 50 milligram of metal complex and 40 microliter of sodium borohydrate. It is a reducing agent, and 100 micromolar of para-nitrophenol and distilled water, double distilled water is used as a solvent. After mixing together, and the UV spectrum was recorded with various time intervals, and you can see here the this I will take the okay. So this is the para-nitrophenol, and the peak is observed at 400 nanometer. After a particular time, you can see that the absorptions of para-nitrophenol peak is gradually decreased, and the new peak is formed. It's around 310 nanometer, which is corresponds to the para-aminophenol. And the isobatic point is clearly stated that there is no side reactions or side products are formed. After 70 hours, the compound is completely decomposed to the para-nitrophenol and the para-aminophenol is formed. And this percentage of conversion has been confirmed by gas chromatographic mass techniques. The gas chromatographic mass spectroscopy was recorded after 70 minutes of reaction sample, confirmed that 100% conversion of para-nitrophenol into para-aminophenol based on the molecular weight. You can see the molecular weight of para-aminophenol is 109. So the exact peak received appeared in 109. So it's 100% conversion was happened in this reaction. The catalyst is very efficient. And then the reusability of catalyst. So as I mentioned earlier, the metal complexes is easy to recollect it by simple filtration. We have performed the same catalyst and in 5 times in the same under similar experimental conditions. So the catalyst is stable and reusable in 5 cycles. You can see here in first cycle, the 100% yield, the conversion. The second stage, the yield is a little bit less. It's around 80. After that, all the cycles is maintain the efficiency of the catalyst. And this is the transmission emission microscopy of metal complex to confirm the surface of the metal complex, because the smoothness of surface is very important for the energy transfer process. So the size of the particles is 400 nanometer, and surface is very smoothy. Yeah, that's all. Conclusion. Copper-2 complexes have been synthesized and characterized by various spectroscopic techniques. EPR data indicates the copper-2 adapted distorted scorpionar geometry. Para-nitrophenol catalytic reaction was performed by the complexes. Results showed that complexes acted as a better catalyst. Complex was stable and reusable up to 5 cycles without losing the efficacy. Interestingly, the process is environmentally gentle, universal, efficient, safe, and operationally simple. I would like to thank Professor N. Raman, my PhD supervisor, and Professor S. Madheshwaran, IIT Bombay. He is my postdoctoral supervisor, and Professor Christoph Josvik, Medical University of Lublin, head of the Biopharmacy Department in Lublin, and collaborators. Professor Janet R. Marrow, University of Buffalo, and Professor Edwin Hooker, Laboratory of Colorado State University, USA, and Dr. D. Mahendran, Griffith University, Australia. Financial support, I would like to thank Indian Institute of Technology, Bombay, Department of Science and Technology, Government of India, and Polish Academic Exchange National Program, NAVA. Thank you so much. I would thank you for your kind attention. I would be happy to answer your questions.
Thank you, sir. Participants, if you are having any queries, you can ask.
Sir?
Yes, please.
You told the para-nitrophenol is a toxic pollutant according to Environmental Protection Agency.
Yes.
Why the government is not banned that type of the usages of para-nitrophenol?
Oh, okay. So there is toxicity, but still, it's not. It's a long-term toxicity, not a sudden toxicity. So we are working to decompose the molecule, and we can able to convert as soon as possible. So we have to give some time to judge, right? For example, if something is explosive, government will stop suddenly. But this is not sudden toxicity. It is a long-term toxicity. And yeah.
So, sir, is it possible to use the para-nitrophenol directly as a metal complex before its harmness is reduced?
Can you repeat one more time? Is it possible to use paranitrophenol as a metal complex?
Yeah, actually, we convert the paranitrophenol into para-aminophenol.
Yes.
My question is why we can't directly use this paranitrophenol directly?
Yeah, no, actually I'm using directly only. I'm using paranitrophenol directly, used as and I put some catalyst and reducing agent. I'm just converting paranitrophenol to para-aminophenol. It's one of methodology. So your question is why why we should not use para-aminophenol directly, I guess. You're asking
So
different way.
my question is, after that, the paranitrophenol is converted into para-aminophenol. Then
Aminophenol,
only we
yes.
are using that metal complex
Okay.
as a environmental hazard agent.
Yeah.
Reducing
So
agent.
okay. So the thing is why I am choosing this one. You are asking the question in this way. So let me answer. The thing is the paranitrophenol is present abundantly in water. So if you take the water and if we add the metal complex into the water, for example, industrial wastewater or agricultural wastewater, and we can able to convert the paranitrophenol to para-aminophenol. That is the target actually. So this is just a preliminary study we have done, but further we have to explore some other studies to elaborate it.
Okay. Thank you, sir. Any more queries?
Hello, sir.
Yes, please.
You said catalyst was stable and reusable until 5 cycles.
Yes.
Have you tried it for the 6th time? What will happen?
6 times I didn't try it honestly, but I hope it will be stable more than 5 cycles. But usually in publication purpose, it's a standard one is 5 cycles. So that's why we just mentioned it in the 5 cycles in the articles. Yeah, but I'm sure it will be stable more than 6 cycles, 5 cycles. Sorry.
Any more queries? Okay. Thank you, sir. Thank you, sir. Thank you for your presentation.
Okay.
Okay, participants, the feedback link is in the chat box. You can fill it and send it. One more announcement. Tomorrow, 10 a.m., that is in Indian Standard Time, you'll have the paper presentation. So you can present your paper at that time. Thank you. Thank you, Praveen, sir.
Yeah, thank you.
Thank you.
Thank you, Anna. Anna, thank you so much for your presentation. It was really nice.
Okay, thank you. Thank you, ma'am.
Thank you. It was a really nice meeting and great honor for us to be invited and be a speaker during that nice meeting. Thank you very much.
Okay, thank you, ma'am.
Yeah.
So participants, you can fill the feedback form. It's in the chat box.
வந்துருச்சா ஃபீட்பேக் ஃபார்ம் chat பாக்ஸ்ல